so as I understand it, you're using Bayesian optimization. You're sort of like, this is an active learning loop where you're choosing sort of the top set of candidates to go and run your assay cascade against, and then feeding the data back into the model. Can you give us? Sorry, go ahead.
Yeah, I explain how that works in outlines. So we've got this synthetic accessibility based approach. Yes. So we
Yes.
have reaction schemes, we have reagents, we have a chemical space that we can build, and so as we generate data, we start to be able to associate that data with particular structures in that virtual space. Yeah.
Right.
And so then so the way we approach it, so in contrast to the classical way where you've got this enormous space and you're just picking molecules out of that space that are in some ways already defined, what we're doing is we're searching, if you like, the reagent space. What combination of reagents is associated with the best activity? Yeah. So that's really what the Bayesian optimization is doing, to focus on those patterns of reagents that lead to the molecules that are associated with high levels of activity. Yeah. So there's a Gaussian optimization process, and Gaussian process model and the Bayesian optimization help as we build the data to focus on those regions of chemical space where the activity seems to lie, and those then generate the particular examples of the virtual library which we make, and then of course we test those, and then we get more data into the model and iterate in that way.
Yeah, and my understanding is that one of the benefits of the Gaussian process modeling is that it gives you sort of a measure of uncertainty that you can use as part of navigating the trade-offs between exploration and exploitation of the knowledge that you've gained. Can you give us a sense of sort of how uncertainty is measured inside of the process that you just described?
Uh, it's actually quite tricky to do in a sentence or two. Um, but I guess we're looking at the sort of nearby reagents, and how likely it is that this would lead to an activity that's within a certain level of what we already know. It's actually quite hard to quantify because of the discrete nature of what we can make. But I think, I think we get a pretty good idea if the signal is pretty flat and not very strong. It's certainly not at the level where you have very, very high degree of confidence in any of these predictions. There's certainly lots of false predictions, but it's good enough to make progress. And I guess at this current stage of development of these methods, I guess that's all we can hope for at the moment. It can, and we have found interesting improvements in the structure through these sorts of approaches. And there's many ways which we are in which they can be approved, and we're trying to approve them. But sorry, I mean that may be not a very clear answer to your question.
No, no. I know that
But that's
there's
sort of where
I
we
know
are.
that, yeah. No, that makes a lot of sense. That makes a lot of sense. I think for, I mean, Gaussian process modeling has been around for a very long time, and sort of deep learning is all of the rage right now. And I know that you had a 2023 paper in the Journal of Molecular Modeling with Ravneet Jagdev and Thomas Madsen, where you took the Stokes et al. Hallison paper apart in a specific way, and you showed that Hallison can be recovered from the same data set using methods that are a lot older than deep learning. And so, I'm interested in that result there, and what conclusion you come to about where you want sort of these data hungry methods like deep learning in a drug discovery process versus where these sort of tried and true methods that are older and maybe and sort of more principles based are the right approach.
Yeah, yeah. I think it really comes down to validation of the methods. The many of these generative AI or deep learning methods, are very complex. They're using a lot of computer power, and they're generating models with very very large numbers of tunable parameters, and so on. And it's and they can produce results that superficially look extremely good. How publication on Hallicit is one example of that, but there are many others where with completely different approaches to deep learning or generative AI, where the publication seems to indicate that what the method has actually learned. Going back to what we were talking about before in terms of binding, the method does seem to have learned the physics of the interaction of the reaction between the ligand and the protein, because in all their validation they got very high correlations with the data sets that they tested. However, subsequently it's proven that those results were artifactual, and were arising from something else, and those something else's are frequently in the preparation of the data. There are many ways in which these sorts of the chemistry data sets can be biased, and that just improves the chances of getting a false correlation, for example. So these things are very difficult to do, and I think looking at and the data generation of a good quality data set is difficult to do, but also one thing which they didn't do in the Hallicit paper, and I think is frequently lacking, is benchmarking it against a really simple methodology. There are lots of simple methodologies that could be applied in most cases, and it's really the improvement you get over those simple methods that are telling you to what extent all of that extra complexity and sophistication and computer power are really doing anything useful for you.